Okay, so this is going to be the fourth and hopefully final recording of the digestive tract, and we're going to go over the pancreas, liver, and gallbladder, which we refer to as the accessory structures. So the pancreas, liver, and gallbladder are accessory organs. Like salivary glands, these are exocrine glands that secrete their product through the duct to the outside of the body. Um, they secrete their products into the GI tract and they appear within the body, but the alimentary canal is open and it's technically considered outside the body, as I mentioned, the lumen of the GI tract. This includes the lumen of the stomach and the lumen of the small intestines. Thus, the lining of the GI tract is considered external body surface. So the first one is the pancreas. This is a gland with both endocrine and exocrine functions. Endocrine functions are going to secrete the hormones insulin and glucagon, which affect most body cells. Exocrine secretions, these are going to be enzymes secreted primarily by clusters of acinar cells. They get released into the ducts of the pancreas that empty into the GI tract. The pancreas is primarily in the left upper quadrant of the abdomen and extends from the duodenum to the spleen, and it is composed of three retroperitoneal regions: the head, which is the wide section in contact with the duodenum; the body, which separates the head from the thinner tail, which tapers off towards the spleen. You also have the main pancreatic duct, which travels down the middle of the pancreas and receives the secretions from the acinar cells. Near the duodenum, the main pancreatic duct merges with the duct from the liver and the gallbladder and enters into the duodenum. Now, each acinar cells are going to be in clusters referred to as acini or acinus, and this will surround a small duct into which the cells secrete their products. Most of these small ducts will merge and drain into the main pancreatic duct, although some can actually secrete into a smaller accessory pancreatic duct. And here you can see the pancreas, you can see also the main pancreatic duct, and then you can see how it merges with the common bile duct coming from the liver and the gallbladder, and then we'll eventually enter into the duodenum there. The pancreas is going to secrete pancreatic juice. This is collectively the secretions of the pancreatic acinar cells and the duct cells, and consists of water and multiple digestive enzymes and other proteins. The duct cells will also secrete bicarbonate ions to make pancreatic juice alkaline. The alkaline juice helps to neutralize the acidic chyme that enters into the duodenum from the stomach, and it protects the duodenum from damage by the acid that's being produced. The digestive enzymes secreted by the acinar cells are crucial in chemical digestion because they catalyze the reactions that digest carbohydrates, proteins, lipids, and nucleic acids. The pancreatic secretion occurs at a basal rate between meals. During eating, pancreatic secretions will rise due to parasympathetic and hormonal stimulation. There's a hormone referred to as CCK, or cholecytokinin, which is produced by the duodenal-enteroendocrine cells in response to the presence of lipids and partially digested proteins in the duodenum. The more lipids and the more partially digested proteins you have in the chyme that's being put into the duodenum, the more CCK you will release. Okay? And the reason for this is CCK will actually then tell the pancreas that you need lots of pancreatic juice because you have lots of lipids and partially digested proteins. Secretin is released by the duodenal cells in response to the acid and the lipids in the duodenum, and this primarily triggers duct cells to secrete bicarbonate ions. So essentially what you have going on here is, depending on the substances that enter into the duodenum, the chemical composition, that will then trigger the release of these hormones, which will then act on the pancreas to secrete their secretions. Okay, so the liver and the gallbladder. The pyramid-shaped liver is located in the upper right quadrant of just inferior to the diaphragm surface. The gallbladder is actually a small sac on the posterior side of the liver. It's close in anatomical and functional relationships. It's actually nestled underneath one of the lobes of the liver. So the liver is covered by a thin connective tissue capsule and visceral peritoneum, and is composed of 4 lobes. The large right and left lobes on the right and left sides of the liver, and is separated by a fold of visceral peritoneal referred to as the falciform ligament, which also anchors the liver to the anterior abdominal wall. And here you can see the 2 lobes here and this big falciform ligament right there. The round ligament on the liver's inferior surface is a remnant of the umbilical vein, which is present in the fetus. The smaller caudate lobe and quadrate lobe are on the posterior side of the liver's right lobe. You have the porta hepatis. This is an indentation on the liver's posterior side where numerous blood vessels enter and exit the liver. This includes the hepatic artery, hepatic portal vein, and hepatic veins. Nerves and lymphatic vessels and the common hepatic duct also enter and exit via the porta hepatis. And this is looking at the posterior side here. We can see the gallbladder, but then you can see this. Here's your vena cava right here. Here's the hepatic vein. Okay. But here's that the porta hepatis right here, where you have the hepatic portal vein, the hepatic artery, and the common hepatic duct. The liver is going to be broken up into liver lobules. These are the basic units of the liver. They are separated from one another by septa, that branch in from connective tissue of the capsule of the liver. They are composed of flattened plates of cells, hepatocytes, that are arranged in the shape of a hexagon and are stacked on top of one another, with a central vein in each of the lobular centers. You have a portal triad at each of the 6 corners of the lobule, which is composed of the following 3 structures: a hepatic arteriole, a portal venule, and a small bile duct that carries bile. The hepatic arterioles and the portal venules both drain large leaky capillaries called hepatic sinusoids that pass between the rows of the hepatocytes. The sinusoids have walls that are not continuous and allow large substances to enter and exit from the bloodstream. Blood flows slowly through the sinusoids as materials are exchanged between the blood and the hepatocytes, eventually draining into the central vein. Central veins then merge and drain into the hepatic vein. In turn, feed into the inferior vena cava. Bile flows through the liver lobule in the opposite direction, from the hepatocytes into tiny ducts called bile caniculi, and will eventually drain into the bile duct. So here you can see you have one of these liver lobules. And if you look, you can see you have the bile duct here, but then you also have the hepatic arteriole and the porta venule. And what's going to happen is blood's going to flow through here, and it's going to go through these hepatocytes, which will detoxify anything, remove nutrients for storage. Essentially, your liver's processing everything that was absorbed in through the small intestines. And then once that all occurs by these hepatocytes, which are working very hard to process all those nutrients or detoxify anything, the blood then gets returned into this central vein here within this lobule. As bile is produced by these cells, though, it gets dumped into this bile duct, which will then, eventually go and to be stored by the gallbladder. Bile is produced by the liver, but it's stored by the gallbladder. So the functional liver, the liver has one of the most diverse sets of functions of any organ in the body. It's actually going to release both endocrine and exocrine secretions and converts harmful chemicals into non-toxic substances that the body can eliminate. The main digestive function is to produce bile. This is a liquid that contains water, electrolytes, and organic compounds. And bile serves 2 critical functions. It's required for the digestion and absorption of lipids, as well as a mechanism by which the liver excretes waste and other substances that the kidneys cannot excrete. Bile salts, one of the main ingredients of bile, are derived from cholesterol. They're amphiphilic, which means they have both polar and nonpolar portions and allow them to interact with both lipids as well as the watery environment of the small intestines. What happens is they will coat the lipids in the duodenum and physically break them apart into smaller pieces, which is a process referred to as emulsification, and mechanical digestion necessary for the chemical digestion and absorption of those lipids. Dawn detergent actually does the same thing if you're washing your dishes. Bile also contains varying amounts of material that the liver excretes, including cholesterol, waste products, and toxins such as heavy metals. Most of these products are not reabsorbed by the small or large intestines and so pass into the feces. The hepatocytes will secrete bilirubin, which results from the breakdown of hemoglobin in the spleen into bile. Normal flora of the large intestines convert urobilinogen into sterobilin, which is responsible for the characteristic brown color of feces. Some remains in blood and is excreted by the kidneys and can be removed via the urine, and that's what actually gives the yellow color to urine. The liver also performs a host of other functions, including nutrient metabolism, which is going to process nutrients that are obtained from the diet. So all those nutrients that got absorbed into the small intestines will pass through the liver for metabolism to occur, detoxification, all alcohol, drugs, any substances that you eat or drink, which could be harmful for the body, will be detoxified by the liver. And then finally, excretion, which is where it's going to directly excrete bilirubin in bile along with other substances the liver processes, particularly certain antibiotics. It also modifies substances that might be or can be excreted by the kidneys. Now, your gallbladder, this is a small sac that sits in the posterior liver. It receives most bile from the common hepatic duct. It stores bile, concentrates it by removing the water, and then it releases it when it is stimulated by the CCK. This will trigger the contraction of the smooth muscle in the wall of the gallbladder, causing the gallbladder to release bile into the cystic duct. The cystic duct will then join the common hepatic duct to form the common bile duct. This joins the main pancreatic duct near the duodenum at the hepatopancreatic ampulla. The ampulla is surrounded by a ring of smooth muscle, and this controls the emptying of bile and pancreatic fluids into the duodenum. Bile contains calcium, salts, and cholesterol. Both can precipitate and form hard lumps called gallstones. People that have inflammation of the gallbladder can actually have, or have too many gallstones that accumulate in there, can actually have their gallbladder removed, okay? The reason why you can remove your gallbladder and still function without really much disruption to your physiological functions is because the liver produces bile continually, but generally does not secrete it at a basal rate. The secretion occurs in response to gallbladder contraction and the sphincter relaxation. This activity is mostly accomplished by CCK and to the small intestines by the vagus nerve. Other factors influence bile secretions, including the hormone secretin, stimulation of bile production, and release by the hepatocytes. The most potent stimulus for bile production and release is bile itself, specifically bile salts. As bile salts re-enter the liver, bile secretion rises dramatically in a positive feedback loop. The bile secretion continues into the duodenum until the duodenum empties, at which point CCK and secretin levels will decline significantly, and you will stop producing the bile there, okay? So what happens is when you have your liver or gallbladder removed, you can't have your liver removed. When you have your gallbladder removed, that will actually cause a little bit of disruption as far as the emulsification of lipids, because that's where you're storing and concentrating bile for use when you need it. However, you're not cutting off bile production at all. You can still have bile produced by the liver, and that can, meet up and get secreted into the duodenum. What they do tell people that have had their gallbladders removed is to stay away from really, really fatty meals or too high concentrations of fat, because they might not be able to emulsify all those fats, in which case it's going to come out at the other end with feces. Okay, and you can have very fatty feces, which is more of a diarrhea consistency. Your liver, again, if you are, abusing alcohol and other toxic compounds, such as alcohol or drug use, and this is not necessarily recreational drugs, but it could also be prescription drugs. These hepatocytes can actually become inflamed, and they could, due to the inflammation, essentially be killed off and replaced by scarring or scar tissue. And this is, going through a process of hepatitis, which is a flare-up of these hepatocytes, and then being replaced by this scar tissue, which is cirrhosis. If the liver becomes non-functional, because you no longer have an abundance of these functional hepatocytes, that is a big problem for you, because you cannot survive without a functional liver. Liver transplants have come a long way over the past 5 years, in that they now will look at what they call a living donor situation, where they can take a third of a donor's liver if they find a match, and then transplant that into you, and it will actually regenerate to a certain extent, and the donor's liver will actually regenerate back. So this has opened up, the transplants for a lot of people that have that need a new liver. Otherwise, you had to wait till somebody died and then you got the liver transplant. So that was very big for liver transplants. So this just sort of summarizes, again, much like we saw with the GI tract, the different regions. This summarizes all of the structure and functions of the accessory organs, your teeth, tongue, salivary glands, pancreas, liver, gallbladder. Gives you their structural properties, and as well as their functional roles. So, this slide and that prior slide that looked very similar to this are very good to have when you're sort of, really recapping this chapter. And this is the overall big picture of the whole digestive process. Okay. And that is it for the digestive system.